each and every cell in the body has about 20,000 genes. And of those 20,000 genes, about 400 to 500 genes, when they get messed up, we call them mutated, they can lead to changes in the cells. With sufficient amount of time, sufficient number of alterations, these changes could result in the cells completely losing their normal bearings and becoming a cancer cell. The difference between a normal cell and a cancer cell is that the cancer cells keep growing, they break into tissues. Normal cells stay in one place, they only can grow up to a point, they don't move around. And the fundamental alteration in cancer, or what causes cancer, is alterations in the genes. When I say the genes, I want to be very careful. These are genes happening in the adult tissues or the tissues in the body, not necessarily transmitted to their families, their children or grandchildren. And every cancer is a product of altered genes in the cells. And what triggers the changes in the genes? Various things. In some instances, smoking for lung cancer, or exposure to carcinogenic substances in other cancers. In many instances, we don't know what triggers these cancers. It's a natural product of the cellular wear and tear. In the past, when patients came with cancer, we made a clinical diagnosis, we got a biopsy, looked under a microscope, and poured some stains and figured out where they came from, and we call them lung cancer, breast cancer, etc. But today, we don't stop there. We take those tumor tissues, grind them up, open the DNA, map this DNA, and see what genes are altered. Lung cancer, for example, the non-small cell lung cancer, there is a type called adenocarcinoma, cancer starting in the glands of the lungs. And if you look at the microscope, they all look the same, but in about a 15 to 20% of patients, it is driven by a mutation in a gene called EGFR. And in about 2 or 3%, it is caused by a gene called ALK that is altered. They look very different in a microscope, but when you map them, the genes that are altered are very distinct, and the treatment is very, very different. The drug targeting the ALK gene problem will not take care of patients whose cancer is driven by the EGFR gene. So it's really becoming very siloed in that fashion, and the research is progressing along those lines. Chemotherapy and immunotherapy work broadly across various different types, but even there, we are trying to figure out why some patients do better on immunotherapy or chemotherapy than others. Again, the answer is going back to understanding how the genes are altered. And now we have tools and technology to really understand the gene alterations. By the way, the fact that we have 20,000 genes in each and every cell came from this huge project called the Human Genome Project. Washington University was one of the 4 institutions that mapped out this project. It took us about 10 years and $1 billion to $2 billion to figure out the human genome, whereas today we can map out the genes in basically all the 20,000 genes for a few $1,000s. As a matter of fact, very recently, one of the Washington University developed an assay to map out the genes, all the 20,000 genes in a leukemia in patients with blood cancers who are approved by the Medicare. And we are excited about the groundbreaking assay we have developed here at Washington University. So really, things are moving so forward very quickly to really understand what's driving the cancer and targeting them specifically to the extent possible. So today, many cancers, when we have patients with lung cancer or breast cancer, people today map out the genes to figure out what kind of specific therapies they would need. And so I think that's becoming a routine practice in many cancers today.